Hello everyone, and welcome to the last session of the HPP Research Awards. The topic today is food safety and shelf life extension. Hyperbaric as a company was born to give a solution to a food safety issue, and as a microbiologist myself, I feel very proud to present this session. The first speaker we have today is Catherine Rulf. Hi Catherine.
Hello.
Welcome and congratulations to reach the finals of the HPP Research Awards.
Thank you. Thank you very much.
So, the topic you would present today, the title of the session is Acid, Cold, and Pressure Adapted E. coli, Listeria, and Salmonella Invalidation of High Pressure Treated Juices. All right, so whenever you want, you can share your screen and start with your presentation.
Okay, great. Fantastic. So good afternoon, everyone. My name is Catherine Rolfe. I'm a research biologist with the U.S. Food and Drug Administration. As Mario has said, I'm going to be presenting acid, cold, and pressure adapted E. coli, Listeria, Salmonella, and high pressure treated juices. So to begin, looking into high pressure and why this is being used for juices. So in recent years, the U.S. fruit and vegetable juice industry has shown a growing increase in the popularity of minimally processed juices. A frequent technology used for these products is high pressure processing, and it can maintain a more natural product that retains vitamins and minerals compared to traditional thermal pasteurization. High pressure processing, so HPP, is often referred to as a cold pasteurization technique, and this allows for the final packaging of the product prior to the process treatment. So in the image here depicted on the right, this is the high pressure unit within our pilot plant at iFish. So this is a 24-liter unit, and this is manufactured by Avir, and this contains a vertical chamber. So this equipment was used for all the research within this presentation. And so the pressure is applied in a uniform and ubiquitous manner using water as a pressure transfer medium. And HPP, as we know, has a lethal effect on microorganisms, and inactivation is mainly through the disruption of the cell membrane and leakage of internal cellular material. There's also denaturation of enzymes and unfolding of proteins. And so the pressure application is able to maintain and leave those covalent bonds, leaves them intact. And so that can maintain those nutritional and organoleptic properties that consumers really want from their products. And so the main variables which determine the outcome of the processing are the food matrix and characteristics of the product, the pressure level and the hold time that's being applied, the proper packaging for the product, and how susceptible those potential foodborne pathogens are. So we know how the technology works and we know how to apply this on the product, but how do we quantitatively assess the safety for consumers for high pressure juices? So although HPP juices is a novel processing technique, regulations still need to follow the FDA juice HACCP. And so the requirement for the juice manufacturers is to have a 5-log reduction of the pertinent microorganism. And the pertinent microorganism is described as the most resistant microorganism of public health significance that is likely to occur in the juice. So this is not well defined for specific microorganisms or strains or even certain types of juices, especially those types of juices that have various ingredients involved. So typically prior association between juice types and microorganisms are made through previous reported outbreaks or previous research that's being conducted. So in most cases, and within the current research I'm going to be presenting, 3 microorganisms which are a public health concern, are regularly included for high pressure validation studies. And so these are E. coli 0157H7, Salmonella, and Listeria monocytogenes. So the reasoning behind this project was based off 3 main focal points, and this was really to aid in process filings and for those validation studies. First, there's no common methodology for preparing bacterial strains during validation studies. There may be a need to use environmentally adapted cells, as stress adaptations could allow for the increased resistance and may differ between individual strains. Secondly, there's no agreement on the high pressure parameters required. So with no specific combinations for what pressure level and what hold time, would be appropriate for certain types of products. And lastly, there's no consensus on the approach for how to conduct shelf life studies. For example, what time of sampling, what day of sampling, what storage conditions should be maintained, should there be included possible temperature abuse, and really how there's no definition for a straightforward shelf life study. So getting into the project objectives. So the first objective was to do a bacterial screening, and this was to screen bacterial isolates for high pressure resistance. And from there to select a final set of bacterial isolates for validation studies. So really going through multiple strains of each type of microorganism and see the differences within the high pressure resistance. The next objective was to assess the effect of the juice matrices on bacterial inactivation and really look at those high pressure parameters that affect inactivation and where that 5-log reduction is being seen from those selected isolates from objective 1. Objective 3 was to determine impact of recovery procedures on the enrichment of target bacterial isolates. This is following FDA BAM and seeing how those recovery procedures are able to, how they are being influenced by the juice matrices. And then lastly is to define a shelf life analysis for high pressure treated juices and really looking at those sampling dates as well as temperature abuse. So getting into the sample preparation, so this is getting into the bacterial screening section. On the left here, you can see the bacterial isolates that were included. So we have 10 isolates of each for E. coli, Listeria, and Salmonella. And so as you can see here, many of the isolates are from either a juice product or some type of produce-related product, something that could be in the environment of a juice manufacturing or juice processing environment. So really keeping it relative to the product. So from there, we have our isolates. So these were taken from a frozen culture and were prepared in 3 different growth conditions. So we have our traditional neutral conditions, so that's 37 degrees Celsius at pH 7. And our cold adapted, so this is going to be prepared at 17 degrees Celsius in a neutral broth. And that's really to emulate or to simulate the cold environment, which obviously the processing is happening, as well as this is a refrigerated product. So seeing if that cold adaptation could help, could aid in resistance of the isolate. And then lastly, we have acid adapted. So this was grown at 37 degrees Celsius and acid adjusted, HCl adjusted broth. So this is going to be at pH 5. And this is to simulate the, see if there's any added resistance towards an isolate that's prepared, that acid adapted isolate that's prepared this way, seeing if they can maintain themselves within that the low pH environment within these juices. And then from there, these 3 different growth conditions, these isolates that were prepared in these different growth conditions, were inoculated into buffer peptone water at pH 3.5. And so using the buffer peptone water at 3.5, pH 3.5, this is able to mimic a juice product in those high acid environments. So we have the inoculated BPW, and so this is going to be within a vacuum sealed pouch, within a pouch and vacuum sealed. From there it goes to high pressure treatment. So looking at high pressure treatment, preliminary work was done to see where sublethal levels would be for all 3 microorganisms. And we wanted a sublethal level because we want to be able to see the differences within the strains. So from there, 200 megapascals was required for Salmonella and Listeria, and 500 megapascals was required for E. coli 0157. So using those pressure levels, there was initial temperature at 4 degrees Celsius and the treatment time of 180 seconds. So from there, viable counts were taken. They were taken same day of plating was done same day as treatment, as well as 24 hours within cold storage to 4 degrees Celsius, and then following 48 hours in cold storage. So this is to see if there's continued die-off of the cells or if there's any recovery of the cells, seeing how they're able to maintain themselves in that acidic environment. So getting into the results of the bacterial screening. So this is looking at E. coli 0157. We have neutral on the left here, or neutral grown cells, cold adapted and acid adapted. So as you can see, there's a variability between the different strains. We see some that are more sensitive. So you can see in our light blue SEA 13B88, that's a very sensitive strain. It seems to be across all 3 growth conditions. And then you have some that are more resistant. So we're looking at F5853 and TW14359. So you can see there's a variability between the strains, as well as you can see cold adapted seems to be more sensitive compared to the other 2 growth conditions. Similarly, this is for Salmonella. So here you can see there's more similarity between the strains themselves. We do have that one strain of 10C that is more sensitive, but you can also see in here how drastic of a difference between cold adapted versus neutral and acid adapted. So for the Salmonella, the cold adapted growth conditions did not prepare the cells well for a high pressure treatment in the acidic BPW. And lastly, we're looking at our Listeria. So here you can see it kind of gives a similar results as the E. coli. We're having variability between the strains, and we're seeing those that are more resistant and more sensitive. So the next section of this was to get into adding the juice into the work. So from here, we used apple and orange juice, looking at a pressure range of 200 to 600 megapascals, keeping with the hold time of 180 seconds, and started keeping the initial temperature of 4 degrees Celsius. So this juice was purchased from a grocery store, a local grocery store, so it was already pasteurized. So for these juice studies, resistant strains from the previous screening were used, as well as sensitive strains. So you can see those listed on the left there for each microorganism. And so they were prepared in the same conditions, so same growth conditions for neutral, cold adapted, and acid adapted. And from here, we did pressure treatments of 200, 300, 400, 500, and 600 to really see that range and see if there's a certain point where high pressure is more lethal and where we're reaching that 5 log reduction. And these were also continued for plating at same day, 24 hours cold storage, and 48 hours cold storage. And so I'm not going to go over all the results for all three of them because there's a lot of information, but I just want to focus on some ones that really stood out from the data. So this is looking at apple juice. This is E. coli 157 strain. So that resistant strain is in dark blue and our sensitive strain is in light blue. So a top row here is the neutrally prepared and the bottom row is our acid adapted. So as you can see here, as we go through our sampling or analysis time, so this is going from same day analyses to 48 hours on the right, you can see there's a pretty drastic difference between the sensitive and resistant strain. We're seeing about a 2 log difference from our sensitive and resistant strain for our neutrally prepared, and about a 3 log difference from our acid adapted strains at the second row here. So really showing that it's really important to have the appropriate strains being used for this because some will give a inaccurate representation of your processing treatment. Similarly, this is an orange juice. We're seeing again our sensitive strain and our resistant strain are having a significant difference with the results here, especially at our 48-hour time point. So really just showing again that apple and orange juice have the same result. We are seeing a difference between our 2 different strains. And so going into looking at our matrices, so our top row here, this is a comparison between our buffer that was originally used for bacterial screening, apple juice and orange juice. So you can see here in our left column, we have our neutrally prepared and then our cold adapted and acid adapted. But you can really see there's a big difference between the matrix that these are being inoculated into and high pressured. So you can see for a BPW in the gray here, we're having substantial amount of die-off, especially after 48 hours. And comparing that to the apple and orange juice, there's a very big difference. So it really does show that there's a protective effect when you're using these juices compared to, a buffer that does not have any, added sugar or any types of titratable acidity that would be compared to a juice. And so looking at the second row here, this is a direct comparison for apple juice and orange juice. And I'm sorry, this is using E. coli 0157, our resistant strain. And you can see here, there's not much of a difference between the 2 juices at the 600 megapascals. You do see a slight difference in the 48-hour sampling for our acid adapted. We're seeing more die-off from our apple juice compared to the orange juice. And this could be a result of the pH levels or something within the juice themselves. Apple juice was around 3.5, 3.6, and the orange juice was around 3.7, 3.8. And then lastly, I just wanted to go over our shelf life analysis. So as you can see here, there's sampling days up to 75 days. Both quantitative and qualitative assessment was done for these samplings. Temperature abuse was included twice within the sampling within the shelf life. So at day 3, temperature abuse was done. This is really to mimic the product being delivered and maybe possible time in between placing on the shelf. And then at day 12, this is to mimic, a consumer purchasing the product, and maybe there's some delay in putting it into the refrigerator after purchasing. And so getting into our shelf life studies here, so looking at the 75-day shelf life, this is for E. coli 0157H7, and we continued with the resistant strains for our shelf life analysis. But here we're able to see a 5-log reduction reached after day 4. And what's interesting with this is that performing the qualitative, our enrichment with this, we're getting negative results. However, we're getting plating results. So, why are we getting this unsuccessful recovery? It could be a function of the enrichment does not work well with the juice matrix. Maybe there's something in there that's blocking the recovery of the cells, or maybe acid adapted needs a little bit more time for recovery during these enrichment processes. And again, this is for E. coli 157 in orange juice. We're seeing similar results where we're getting negative enrichment, but positive plating. But just to reiterate, we were seeing a 5-log reduction. You're able to see that with the plating, and that was reached at day 4. And then following up with our shelf life, we have Listeria monocytogenes in orange juice. And so with this, we're not getting any plating results. 5-log reduction was reached same day as high pressure treatment, but you are seeing a positive enrichment throughout the 75-day shelf life. And what's interesting with this, in the tables on the right here, you can see at day 35, this is just the amount of time for the enrichment. It took a 48-hour enrichment for positives to show there. And then as we get to 75 days, more positives are showing up within that enrichment. So is Listeria able to maintain itself within this acidic product because it's acid adapted, or is that help aid in being protected from the cold environment? So interesting results for the Listeria. So just getting to a project summary here, E. coli 0157 exhibited significant barotolerance, so it was more resistant to high pressure compared to Salmonella and Listeria. Juice matrices provided more protection of bacterial cells compared to the buffered peptone water. So we did see those significant differences in the recovery, even up to the 48-hour time point. And so the results of this research propose a barotolerant and acid-adapted strain of juice-associated pathogens should be used in a cocktail inoculation for validation studies of high-pressure treated juices. So really making sure we're having appropriate strains that are being used and making sure they're not overly sensitive or overly resistant to the high-pressure process. And then lastly, a pressure treatment of 586 megapascals achieved a 5-log reduction of E. coli 0157H7 after 4 days in cold storage for both juice types. And so this is indicating a cold holding time is necessary for these high pressure treatments. It could be depending on the juice types, but from this research, we were seeing in both types of juices, the same amount of cold holding was necessary. And with that, I'm happy to take any questions. And then my email is listed there as well if anyone wants to reach out.
Thank you, Catherine, for your presentation. It was really nice. I think your work really tries to give an answer to a question that the juice industry needs to find.
Mm-hmm.
So yeah, I have a couple of questions, if I may ask.
Yes, of course.
Yeah, because I saw in your results that doing the cold adaptation made the strains more sensitive to HPP. So I was wondering if it would be a good practice to cold adapt strains for process validation and skip acid adaptation.
Right. So you mean if you do cold adaptation as well as acid adaptation?
I mean, because the food industry is a cold environment, so some companies may think that doing cold adaptation is just enough.
Right. Yeah. So looking at the cold adaptation results, it was showing to be more sensitive across all three microorganisms. This could be a function of that cell membrane. The cells try to balance out their membrane fluidity, and with the cold adaptation, it could make it more susceptible to pressure. So just from the results here, I was having more resistance shown from the acid adaptation. Some have asked, what if we were to cold adapt and acid adapt? I have not tried this. It could perhaps, add a little bit too much stress to the cell. But just from the current results, I was definitely seeing that the acid adapted was a little bit more resistant than the cold adapted growth conditions.
Yeah, interesting. Thank you.
Yeah.
So maybe one more question. Do you think that the 3 pathogenic species that you studied are relevant for process validation, or you found that maybe some species are more sensitive and can be, let's say, avoided in process validation?
Right. So the 3 that were included are the ones that are regularly included for validation studies, and those are the ones that we're most concerned about, most likely to occur. I was seeing E. coli 0157 was much more resistant than the other 2 microorganisms, but it is good to include all 3 to make sure, just in the juice products that I was using, that was the case, but depending on the certain ingredients and certain pH levels, each product's different. So I think more work would be needed to see, specifics if other types of species would be more relevant. But for the current validations, I think that all 3, it really covers the basis. So it was interesting for the work here as well.
Yeah, I understand. Thank you.
Yeah.
And yeah, maybe last one to wrap up. What is the pending work to achieve a safe harbor for HPP in the food industry, in your opinion?
Right. So I definitely think more work, looking at different types of juices, different characteristics in there, and really to see, how that influences. As well, I was using single strain, so it'd be interesting to see, in a cocktail inoculation with these various, resistant strains, is there any interaction within there? So it'd be, good to look and see how those are influencing the high pressure process as well.
Yeah, thank you so much.
Yeah, of course.
So from my side, it was all. I don't have more questions, and I think we can continue with the next presentation.
Okay, perfect. Thank you so much.
Great. So let's continue with the HPP Research Awards in this session on food safety and shelf life extension. Right now we have here Elizabeth Burneman. Hi, Elizabeth. I hope I pronounced well your name.
Close enough.
So yeah, thank you for joining us and congratulations for reaching the finals.
Thank you.
And yeah, so today you will present us your work entitled High Pressure Processing of Heat and Pressure Resistant Fungi as affected by pH, water activity, sulfites, and dimethyl dicarbonate in diluted apple juice concentrate. Okay, so the floor is yours. I will let you share your screen right now so you can start the presentation.
Thank you. So yes, the title of my presentation is High Pressure Processing of Heat and Pressure Resistant Fungi as Affected by pH, Water Activity, Sulfites, and Dimethyl Dicarbonate in a Diluted Apple Juice Concentrate. Quick background about myself. I hold 3 degrees from Cornell, concentrating mostly in food science. And I've also worked at Unilever, both before and after graduate school, for about 5 years total, supporting Skippy Peanut Butter, the spreads business like I Can't Believe It's Not Butter, and Country Crock, and more recently, the ice cream business across all ice cream brands. So today we'll start by discussing the background and the objective of this study, followed by the methods and the overall experimental design, and finally the results. So we all know that HPP juice is a large category, and this is because raw juice may only be sold directly to the consumer. Otherwise, per FSMA, it needs a treatment to cause a 5-log reduction of pertinent pathogens. So there's a fair amount of information available on the safety of HPP and reduction of pathogens in juices, but not necessarily on spoilage or shelf life. And this is a concern because we know that bacteria and fungal spores are highly resistant to HPP. In fact, HPP can act as an activation step, causing germination in the final product, leading to unwanted clouding and visible growth, as well as off odors and flavors. So primarily, products are kept refrigerated after processing to prevent this. However, this can be quite expensive. We also know that the efficacy of HPP is medium dependent, with many studies having been done with benchtop models, but these results may not necessarily translate directly to commercial units, or studies have used buffers, not targeted foods or inappropriate organisms. So if these organisms are so resistant, how can we control them? We can use hurdle technology and combined methods to have a synergistic effect that results in an extended shelf life. So we can use high pressure, water activity, pH, refrigeration, and there's potential to add preservatives. Of course, preservatives tend to not be very clean label friendly, but there are a few options available. Dimethyl dicarbonate and sulfites are preservatives commonly used in beverages, most commonly wine. Presence of DMDC cannot be detected within hours of treatment because it breaks down, which results in a clean label preservative. And sulfites do not need to be declared if they're used as a processing aid, and the concentration is less than 10 parts per million. So the overall objective was to evaluate the use of high pressure processing to cause a reduction of heat and pressure resistant spoilage fungi, and then to look at the effect of water activity and pH to see if we can get a synergistic effect. And finally, to test the effect of adding processing aids to see if we can make that even greater of a reduction or an extension of shelf life. For experimental design, we started by growing fungi in malt extract agar for 30 days at optimal temperatures and then harvested the target organisms. We also adjusted apple juice concentrate to water activities 0.94, 0.96, 0.98, and 1, and pHs 3.5, 4.6, or 7. In some cases, 8 parts per million potassium metabisulfite to stay below that 10 part per million declaration requirement, or 250 parts per million dimethyl dicarbonate as a proof of concept to see if there's a synergistic effect with HPP. All samples were processed at 5 degrees Celsius, some at 450 megapascals for just 1.5 minutes, and the rest at 600 megapascals for 15 minutes. All samples were stored for 6 months. Spoilage organisms were chosen based on common occurrence in spoiled foods. So Paecilomyces niveus can cause spoilage of heat-processed acid foods and can even grow in low-oxygen environments. Aspergillus pseudoglaucus is quite xerophilic and has been known to cause spoilage in a wide range of foods from fruits to cheeses and dried meats. And Aspergillus fisheri has been known to cause spoilage of fruit products and is extremely heat-resistant. Now you notice that there are 2 sets of names for each of these, including Pisciclamys, Xerotium, and Neocytoria. Since 2014, the mycology community has been working on renaming and simplifying the names of these organisms based on phylogenetic information rather than morphology. So papers older than 2014 may be using these older names, but they're most likely the same organisms. As far as overall framework, breaking down all the pieces and conditions tested, starting with pH 4.6 here in the middle, that was chosen as a midpoint because it inhibits the growth of Clostridium botulinum. We started by processing at 450 megapascals for 1.5 minutes. This was largely ineffective, so all other conditions were tested at 600 megapascals for 15 minutes, and samples were stored refrigerated. In some cases, as a proof of concept, 8 parts per million sulfites were added to samples, and these were stored ambient to test survival. Moving to the left in yellow, pH 3.5 was chosen to represent apple juice, which has a wide pH range from 3.3 to 4. These were processed at 600 megapascals for 15 minutes and stored both refrigerated and ambient. And then on the other end of the spectrum, pH 7 was chosen to observe the effect of HPP and activation kinetics at a neutral pH. These were processed at 600 megapascals for 15 minutes, stored refrigerated and ambient. And then as a proof of concept, we added 250 parts per million DMDC to Aspergillus fisheri and stored this for ambient to test survival only. For results, starting with Aspergillus pseudoglaucus here, on the y-axis you'll see log reduction, and on the x-axis you'll see water activity. Overall, you can see that reduction of Aspergillus pseudoglaucus increased as the water activity increased. You can see with the red line at the top, we achieved the highest reduction with pH 3.5 at 600 megapascals for 15 minutes. Just below that, with the dark blue dotted line, you'll see pH 4.6 with sulfites and pH 7 without any additive, both processed at 600 megapascals for 15 minutes. And if you notice, the dark blue dotted line had significantly higher reductions of Aspergillus pseudoglaucus than the dark blue solid line. Both of these were pH 4.6 processed at 600 megapascals, but the sulfites were significantly better at reducing Aspergillus pseudoglaucus. Finally, worst case scenario was the 450 megapascals for just 1.5 minutes. As far as storage ambient storage showed visible mycelia of Aspergillus pseudoglaucus pretty quickly, even with the sulfite. So even though we were able to cause a greater reduction, the sulfite was not particularly helpful at extending shelf life. However, with refrigerated, we were much better. And with the exclusion of the lower pressure, where we saw visible mycelia pretty quickly at 6 weeks, we were able to get to 17 weeks at the lower water activities and 20 at the higher water activities for pH 3.5, and then similar results with 20 and 26 weeks achieved at pH 4.6. No growth observed for the full 26 weeks. This is most likely because the optimal growth range for this organism is around 4.5, so the higher pH was actually acting as an extra hurdle. Next, we have Aspergillus fisheri, and you'll notice with these crisscrossing lines, the pH differences were less distinct. And you'll also notice these lines are quite flat, so we lost that water activity effect. So with the red line, again, pH 3.5 processed at 600. The green line, pH 7 processed at 600. And the light blue line is actually pH 4.6 processed at 450 for just 1.5 minutes. We were able to achieve a 1 log reduction of Aspergillus fisheri. With the dark blue solid line, pH 4.6 processed at 600 megapascals, there's largely not much of a difference of post-processed counts as pre-processed. And then down here, you'll see with our processing aids, we actually had an activation of the spore, so we had higher counts after HPP. So the green dotted line is pH 7 with 250 parts per million DMDC, and the dark blue dotted line is pH 4.6 with 8 parts per million sulfites. So these caused a 1 to not quite 2 log increase. As far as storage time, again, visible mycelia was quite quick at ambient, but thankfully there was no growth observed for 26 weeks at refrigeration, even though we saw activation or minimal reduction under the conditions tested. Finally, Paecilomyces niveus was the most pressure-resistant species tested. So note the 0 is quite high on the y-axis, and the majority of the conditions tested showed activation instead of reduction. And also the dotted line showing sulfites had little effect. For ambient storage, it was again quite quick to visible mycelia, with one exception. With the addition of sulfite, even though we didn't see significantly higher reductions when adding sulfites, there's no visible growth with the 0.94 water activity condition for the full 26 weeks stored at ambient. So the sulfites had a really limited effect on the other 2 organisms and at the higher water activity tested for Paecilomyces niveus, but under this one condition, it did appear to be helpful. As far as refrigerated storage, again, no growth was observed for 26 weeks, which was really important because this organism was highly activated by HPP, but thankfully it was unable to grow under refrigeration. How do our results compare? Obviously, not everybody's done exactly the same thing that we did, otherwise we wouldn't have a very interesting study, but there have been some studies looking at 600 megapascal processing, although not for as long of a time period, and they've used buffers, mango purees, sucrose solutions. These studies also found that these organisms, or very similar organisms, were highly pressure resistant. They were only able to achieve 1 to 2 log reductions, or they saw germination. For conclusions, a 17 to 26 week storage period could be achieved, but only at refrigeration. Paecilomyces niveus was the most resistant to HPP, while Aspergillus pseudoglaucus of the 3 organisms tested was relatively sensitive, but was capable of growth at 5 degrees Celsius. So Aspergillus pseudoglaucus is probably going to be your target organism when determining HPP parameters. The addition of sulfite or DMDC did not significantly improve shelf life extension after HPP, except in one condition tested. Overall, Aspergillus pseudoglaucus, Aspergillus fisheri, and Paecilomyces niveus were very pressure resistant, even at pH 3.5, 600 megapascals for 15 minutes. But again, refrigeration was key in extending shelf life. For final processing recommendations, definitely would recommend going up to 600 megapascals for 15 minutes. There's a slight possibility of adding sulfites due to the fact that we did see a longer shelf life for Paecilomyces niveus at the lower water activity, but it's a little unclear how helpful that is since it actually caused activation of Aspergillus fisheri. So there's a potential to focus more work on looking at the sulfites, maybe looking at lower water activities, or we only tested the effect of sulfites with an ambient shelf life, so it could be helpful with a refrigerated shelf life. So for future work, we could look at the sulfites, we could compare directly the storage conditions. There's also potential to increase processing temperatures, which would add an extra hurdle to get more of a synergistic effect. Could also try cycling pressures. Other studies have found that pressure-resistant organisms can take up to 5 cycles of causing activation of those spores and then inactivation. So that might be the last choice for future work just because it would be so time-consuming to process over and over again these products. I'd like to thank the National Institute of Food and Agriculture for funding this project, as well as my advisors and the labs where I completed this work. And I will take any questions.
Thank you so much for your nice presentation, Elizabeth. I think it was very clear. So yeah, maybe I have written down here a couple of questions just for the audience to have some concepts clear. You mentioned that you worked with pressure and heat resistance fungi spores. So I was wondering if you found any correlation between the heat resistance and the pressure resistance of each species.
Yes, so the heat-resistant organisms tend to also be very pressure-resistant. So these tend to have really thick ascospores. Some of these have sexual reproduction, which makes them even tougher than some of your asexual reproductive spores. And so because of these physiological ways of evolving, they are resistant to both. They tend to be.
Okay, now it's clear. Yeah, maybe a question to for people working in the juice industry, yeah, so they can sleep at night because yeah, some of your results were a bit scary. So I was wondering, how would you explain to someone in the juice industry your main findings? Should they be okay, or they need to change something?
So I would say one of the hopes initially when exploring this project was that maybe under some conditions, refrigeration wouldn't be so important. But we confirmed that yes, you do indeed need to refrigerate your juice, which is key. And so that would be very important. Secondly, we were able to cause pretty good reductions of the organism that's capable of growth at refrigeration. So that's very helpful. I would target Aspergillus pseudoglaucus as the target of concern, because the other two, while very pressure resistant, were unable to grow at refrigeration.
Yeah, I agree with you here. It's very important to keep products chilled, so you prevent growth not only of fungi but other species.
Mm-hmm.
So yeah, I think this was all from my side. I had more questions, but you already answered them during the presentation. So yeah, I think we're fine.
Awesome, thank you.
Thank you. And yeah, you can keep watching the next presentation of this session. Bye-bye. So we continue with this session on food safety and shelf life extension of the HPP Research Awards. And now is the turn of Javier Rocha. He will present his work on Bacillus cereus spores and Staphylococcus aureus vegetative cell inactivation in human milk by high pressure processing. Hello, Javier.
Hello.
I hope you are ready.
Yes, I'm ready.
So whenever you want, you can start with your presentation.
Okay, thank you. Hi, my name is Javier Rocha Pimenta. I am a PhD student in the Center of Scientific and Technological Research of Extremadura, Spain. And I am going to present our study. I hope that you enjoy it. Consumer concern for food safety has increased, and for this reason, the agri-food industries invest a great deal of resources on the processing and production of healthy and safe food. From a microbiological point of view, the development of safe food includes the analysis of the raw material and other materials that intervene in its manufacture process. Human milk is recognized as the best option for newborn nutrition, as it provides them the needed nutrients for the development. Furthermore, human milk includes a wide variety of immunological factors that prevent the development of present and future disease. It is important to ensure milk safety due to the precarious health conditions sometimes presented by infants. The pasteurization is the most utilized method to assure pathogenic microorganism-free milk. A low-temperature, long-time thermal pasteurization, named the Holder pasteurization. The problem of this applied technology for milk pathogen elimination is the elimination of nutrients and other important elements of the product during processing, which led us to research safer and more efficient alternatives. At present, one of the processes on the rise in the agri-food industry for the control of microorganisms is high-pressure processing. High-pressure processing treatments cause critical damage in microorganisms found in the foods. In the slide, we can appreciate the damage caused by high pressures according to their intensity at different cellular levels. Despite this, bacterial spores are highly resistant to pressure, and it is necessary to apply pressure higher than 1000 megapascals at room temperature to inactivate them. However, the agri-food industry is limited to applying treatment of 600 megapascals, since it uses water as a pressure transmitting fluid. Therefore, the focus has been on the application of high hydrostatic pressure to force spores to germinate and afterwards eliminate them in their vegetative form. Specifically, in the case of the effect of high pressures on human milk compared to other technologies, we can obtain three main benefits: food safety, nutrient quality, and consumer approval. Therefore, the objective of this research were to investigate the effect of HPP treatment against Staphylococcus aureus and Bacillus cereus on human milk, and to generate a response surface model by design expert software, thus optimizing the condition for the inactivation of this bacteria. The effect of treatment and surface response of Bacillus cereus was established in vegetative cell and spores to determine its complete inhibition. Human milk samples were obtained from healthy volunteer mothers from a human milk bank of Merida, Spain. All milk donations were stored until the experiment was performed. Prior to inoculation, the human milk samples were sterilized by autoclaving to eliminate initial microorganisms. Staphylococcus aureus and Bacillus cereus were inoculated to provide a standard concentration. For Bacillus cereus, inoculation of spores, sporulation was checked daily by microscopic examination and heat treated to eliminate vegetative cells. Thermal processing was applied in the sterile glass tubes with 10 milliliters of milk. For the study of the effect of hydrostatic pressure, the milk was treated in a semi-industrial discontinuous hydrostatic pressure unit, concretely hyperbaric wave 6000 model with a capacity of 55 liters. Staphylococcus aureus counts were determined by Parker agar, and determination of Bacillus cereus counts were determined on agar MYP. We use design expert software for mathematical modeling. HPP treatment was modeled using response surface methodology with central composite design. Two parameters were selected as variables: holding time and pressure. As I said before, the equipment we have in our research center is the hyperbaric wave 6000 model with a capacity of 55 liters. These tables show the reduction of Staphylococcus aureus and Bacillus cereus in human milk after HPP treatment. It also showed the pressure and holding time condition of for HPP treatment proposed by the central composite design. The condition proposed by the model generate maximum count reduction in Staphylococcus aureus and Bacillus cereus respectively. In contrast, whole pasteurization assays on human milk showed a smaller reduction in both bacteria. The most effective treatment in our model was the highest pressure intensity and an intermediate holding time. These results show that HPP treatment are more effective than whole pasteurization to ensure food safety for newborns. Initially, in this study, the analysis of the influence of HPP treatments on Bacillus cereus spores was considered, but this did not conform to the reduction model under study. Data not found. The bacterial spores are activated and germinate when subjected to HPP treatment, becoming vegetative cells. In this way, subsequent bacterial reduction occurs, but depends on the time of germination of the spores and the condition of the treatment, which means that it cannot conform to the response surface model that was intended. Experimental design combined with response surface methodology was used to optimize the experimental variables that have a significant influence in the process. A central composite design was used in this study to screen the importance of two parameters, holding time and pressure, for bacterial reduction. ANOVA results indicate that quadratic model is significant. A statistical analysis showed that the relationship between significant independent variables, pressure and time, and response, bacterial decrease, using a second-order model by response surface methodology can be evaluated. For the model validation, the maximum inactivation in our model has been calculated, resulting in treatment with these conditions. The model gives us this theoretical inactivation, and experimentally, the real results were this, which allow us to consider the model has value. Finally, the graphs show the response surface of the model obtained for Staphylococcus aureus and Bacillus cereus, respectively. According to the results obtained, pressure intensity and holding time have a significant influence on the reduction of their population in human milk. The model shows that a direct relationship between pressure and holding time has variables. Therefore, treatments are more efficient when these two factors increase. As final conclusion of our study, we can conclude that pressure intensity and holding time are important parameters to predict the inactivation of microorganisms by HPP treatments in human milk. Our proposed model was validated, and reduction of Staphylococcus aureus and Bacillus cereus were higher than by whole pasteurization. And the model could not be validated for Bacillus cereus spores, which would probably need a double treatment or the combination with high temperature process. Thank you for your attention, and if you have any question in the slide, you can find my contact email. Thank you.
Thank you, Javier, for your nice presentation. You have very interesting results. So, yeah, I took notes of your presentation. Maybe I have a couple of questions that you can answer for the audience. Yeah, the first one is related to Bacillus, okay, because Bacillus is a spore former.
Yes.
And superdormancy has been reported for Bacillus spores, okay? So yes. I was wondering if you have found this on your research. Maybe this accounts for the yeah, because you couldn't fit the data to a model. I don't know if this has something to do.
No, the spores are the form of the microorganism of resistance to prevent an extreme situation from killing them. In this case, there is a literature that agrees with our data and that has shown that applying 2 successive HPP treatment would kill them. But we know don't do this in laboratory.
Okay, it's fine. So yeah, maybe I have more a general question. Because yeah, this data you have presented show the potential of HPP to preserve human milk. So yeah, maybe I was wondering what are the limitations, the legal limitations to implement this process for human milk banks.
I think that in my opinion, high pressure processing is a very useful and versatile technology that allow us, as I said in the presentation of our study, to ensure the nutritional quality of the product and at the same time to make it safer. But I think that the main inconvenient to the enterprises is the money main. I think it's impossible to enterprise to cost these new technologies if the data is not good and it's safe the economic cost.
Yeah. So you think more research is needed before this is implemented to preserve human milk in milk banks?
Yes. I think it. Yeah.
Okay. Maybe as the last question, yeah, I was wondering how you plan to continue your research, if you have some future steps in mind.
Yes, this is the main objective of this publication is part of a project in which we have several other publications where we have compared other levels, the difference between whole depersonalization and HPP. Part of this study has focused on the difference between the one technology and the other, observing, for example, that long exposure to high temperatures and pasteurization negatively affect immune system factors such as immunoglobulins. I think that this is the future to see how the HPV treatment can affect positively in compare with traditional technologies.
Yeah, I think this is a very complete work. So you not only focus on food safety, but also on quality of the product. Yeah, that's good.
Yeah.
Great. So yeah, I don't have more questions. And your presentation was the last one of this session on food safety and shelf life extension. So I hope you all enjoyed the 3 presentations, and yeah, good luck